large individual cup instead of just sort of a large sprawling colony, which is what you find in tropical shallow habitats.
Yeah, so Pilot, did you copy Amanda's comments there?
I did not.
Okay, the first midwater transects is going to be at 900 meters instead of 800 meters. So you just wanted to make sure you guys up front knew that.
Video's clear.
And if we could take a look at that brittle star in the upper right-hand corner of that same...
Did you copy that, Levi?
Oh, thank you.
It is...
Chris, if I could just say about the cup coral, it might be worth noting that half of all known stony coral species are solitary.
I don't see the other one.
Yes, that's a good point. A lot of people don't realize that a lot of
I don't see it.
Sort of the proper coral diversity is actually in the deep sea rather than in the shallows, which is where everybody sort of presumes a lot of them are.
I can take a partial.
Watch leads. I didn't see the other. Where are we looking?
Your left laser is pointing right where when it was pointing on that rock to the upper right of the coral.
Okay.
Keep
here?
going up a little further and then to the right.
Yeah.
Keep going a little bit more.
Okay.
And swing up the camera and then right from there.
Right
that
here?
little white thing on the black boulder to the right.
Oh, I think it was the layer below it, actually.
It wasn't this?
No, I'm looking at that.
This boulder?
Oh, I'm sorry. Excuse me.
We're not looking for sponges, we're looking for brittle stars.
Sorry.
Sorry to the left.
Okay.
I saw something that looked like it was on the rock face there.
We can look at that also.
Is it this one?
No, it's the one below it. That
this
one.
here. Okay.
Yeah.
Let me get a little closer
Roger.
to it.
Okay.
Well, and then
Lower
I'll
left.
choose
Yeah.
Standing by.
I don't know, weird. Let's see. Let's zoom in.
Oh,
right there.
Now.
Yeah.
Okay.
Sorry.
We got you. I did not see that one at all.
So this is a brittle star in the family Amphiuridae, I think. And these are notorious and ubiquitous. They occur widely, but they're usually very small.
Have a tiny bit more zoom.
And they like to dig into the sediments and the substrates. They're actually quite effective diggers. They're often studied for bioturbation, sort of to look at how they affect the sediment. Brittle stars are like the cryptic, numerous echinoderm group. There are over 2,000, I think closer to over 2,100 species of brittle stars, and their biomass in the deep ocean is considered to be massive, just because they're hidden and present everywhere. And there are videos that show these just sort of digging through different sand layers or sediment layers, and there's sometimes hundreds of them down there, thousands of them. So anyway, thank you.
Yeah, but not here. There's one.
Well, one big one, but the rest of them are all in the sand. And I guess if we can move on from whatever that thing was.
We can.
Unless we really want to look at it.
No, I think it's a dead stump.
Okay.
I just misinterpreted.
No, that's fine. Thanks, Bob.
What's the progress of that move?
Yeah. Background is a Walteria.
Like 3 meters left. Should be about done.
Family Euplectellidie.
Feeling about a 5-minute delay, I think.
Up on it. We've imaged these a couple times today.
Okay, so no interest in this one.
No, not right now. Chris has only had 3 sea stars today.
Oh, we're looking for sea stars now.
Yeah, we're looking for sea stars.
Copy that.
And maybe a few corals and other things too. What the hey? There's a dead vase sponge there. The yellow one. A little weird bamboo
ROV
there.
nav,
Passing off to the left.
Move complete. Thank you, Bridge. Move is complete, pilots.
Can we zoom in real quick on Sirius? Hold on.
Absolutely.
Go ahead.
I see it right over.
Partial.
Go ahead.
Mm-hmm.
You got to snap in.
I see.
I should have been more on the ball, but that last bamboo reminds me of the comment I made earlier that colonies grow in a pattern that reflects the dominant current, but sometimes you can see that they change their orientation. And it looked like that one was growing and maybe tipped over, and so a branch started growing in
yeah.
a different orientation. That's the bamboo coral you referred to.
I'm gonna go look at this and then
I
I'll
see.
head back.
Sea urchin in the lower corner, or in the lower hand side of the screen. It's purple.
Where is it?
Yeah.
Back where I was?
To the right,
To the right.
Right?
and a little bit. There we
right
go.
there. Okay.
Where is
Nice.
it? I don't see
It's
it.
right. I will land.
Oh,
this.
yeah, okay. Good eyes.
Well,
Yeah,
where
nice
spiny
catch.
things are concerned, I have a sense.
Good spotting.
Aha, and we have good stuff to talk about, too.
It's coming video.
Doing.
I'll try to center it up here.
Sure thing.
So this is an aspidodiadematid. Ooh, we've got
2 for
little
1.
stars, too. So this is an aspidodiadematid. This is an urchin that it can actually, so the spines kind of extend from the central body and bow out over to the sediment. And there are actually little walkie pads on those spines. They walk along on the bottom of the seafloor,
Where's
like
your molecule?
kind of like a giant
go ahead.
spider robot. And so these are, it's thought to be related to sort of the diademas that you find in
Anything you wanna look
shallower
on here,
waters.
video? You wanna see?
The characteristic features tend to be
just hold this
actually
for a
very
few more
close to
seconds.
the
Copy.
arrangement of the tubercles of the spines that we're looking at closely here. There's an interesting structure right on the top there. That's, I believe, what's supposed to be called the anal sac. And so that often people think of this as sort of the eye, but that's actually where the poop comes out. And so in a lot of
I'll just take a slightly wider
shallow
shot.
water species, the material comes out and is just sort of jettisoned out of that top area. But if you track one of those spines, we might see that the end of the spines have a little pad on them.
Okay, I think
Let's
we
see.
want to zoom in on
All right,
Yeah.
so the big question
the
here,
spine.
One of the larger ones?
aspidodiadema, plesio. Yeah.
That's a good question, but I think I'll need to look at the close-ups
That
later
might be a
and
better
compare
view for you.
the,
You want to search around video?
compare
Yeah,
the,
the close-up
pra-
just
the
a
pattern
better
of the
stuff.
test, which is the
can see it on
tubercles
this
are
one.
what
Is it the one
where the
this
spines
right
are
here?
connected to the body.
Sure.
Okay.
The other thing that we might be looking at, let's see if we see any of the spine tips in the distance.
Tiny bit up.
Yeah.
Tiny bit up.
But I think that thing, the other
Further,
thing
I
that
think
we're
it's
we might
longer
be,
than I thought.
Yeah,
Oh, this guy.
Oh,
yeah.
The one in the back.
Yeah,
Okay.
perfect.
yeah. Maybe over to the right a little.
We're trying to get one of
Well,
these.
Can't see
he's
what's
looking
on
at
the
this,
tip.
Yeah.
that one there. Is that what
Yeah.
you wanted? Or
Yeah, well, I was trying to look at one that might actually be connected to the bottom, that's in contact with the bottom.
Okay.
Oh, I
for
see.
the hooves?
Let's
Yeah.
see.
Well, they're more like little pads. It's the echinotherids that are usually hoof-like.
These are all pointing up.
And the other thing that's really neat you can see from this image, there are little claw-shaped structures right around the bottom of the central
Exposing.
body there,
We can
which
do
is
that.
what we call the test. And you see they almost look like little alligator claws. Those are what's called pedicelaria. And it's really nice to be able to see one of these alive and with the pedicelaria extended like that. Also present are the tube feet. Those are those long tentacular extensions coming off of the bottom, and that's normally how they would move, or normally how they would pick things up off of the substratum and either remove things from their test or move them to their mouth. But
Okay, video's clear.
yeah,
Okay.
but I think we can sort of move on. That
We can put it
that
in
white
another
structure
ship
next to
movie,
it, I think that might have
Keep
been a
us moving.
brachiopod, a
225 still good?
Yep.
one of those types of animals that we saw
We
the
can
other
just
day,
keep
a very ancient
chugging
group
along.
of
Bridge, are we now?
bivalved animal that are filter feeders.
Bridge,
So
right?
anyway,
Yeah, ready for
interesting
another
and
move if
exciting
you are.
few moments.
sir.
Thank you.
All right, same as last. 30 meters, bearing 225 degrees, speed 0.2.
30 meters, bearing 225, speed 0.2.
It's a good copy.
Coming up.
Negative.
ROV nav bridge, move initiated.
Watchers, any interest on the top of this?
Yeah, why don't we zoom in on the top of that? I think it might be one of Chris's critters.
Sure, let me
Close enough.
Close enough, yeah.
set up for it.
So black coral to the right of it, we've already imaged it. And I think it's a primnoid behind it to the left.
Let's come in video. Balanced on a rock here.
Yeah, so this is a feather star,
Okay, I'll just
and a rather nice one.
hold this from
These are filter feeders. They hold their arms up into the water, and all of these projections that come off of it have in turn themselves small projections. These are called pinnules, and they have little tube feet that come off of them.
Right.
And the water
That's a good
water
bit
current
closer.
carries little food particles, which they use to
Is that all you got?
That's full.
which they
Copy
pick.
that.
They use their arms to pick the food particles out of the water, and they move them down to the mouth. The mouth is that center dark spot where all of the arms are congregating, and then that disc or that little bulb on top is where they actually. That's where the anal vent is, and architecturally very nice because then they can vent off those things away from their mouth so that it doesn't interfere with feeding. These are very old animals, and you'll
Whatever.
see
I'll
there's
tilt that
another
for you if you
form
like.
of this
Okay,
kind of
that'd be
animal that has a stalk, and we'll see
Maybe you
those
can see something on the
at some
top here.
point as well. But
I don't know if
these
that helps you, video.
have lost the
Just a
stalk
tiny bit down.
and can actually swim in some cases. We saw one
Perfect. Hold there.
swimming earlier today. Looks
Yeah,
like there might actually be some more of those funny little frangulose animals or whatever they are on the sponge as well. Interesting.
Yeah, I haven't seen any stock crinoids today, have we?
No. We
That's-
saw another feather star swimming
Whenever you're ready, video.
with that sea
Chris,
cucumber.
I think the pinnules and two feet near the base or near the calyx was swollen there, so I suspect this one is ripe with gonads and ready to
Little
reproduce.
shrimp.
Yes,
Sure.
that's a good point.
Go ahead,
The sort of pinnules, the structures on the arms had large swellings, and those are probably
Take me a bit more.
I'll
so this
try
one
to
probably
track
has
him.
No worries.
juveniles or something,
I'll come wide.
or eggs ready to reproduce. Scott's
thank you.
very good, Scott. Thank you.
So, I'm not.
So we're good to move when you guys are.
Yeah, well,
Copy
I'm
that.
trying
Going
to
up.
figure the shrimp out.
Oh,
Oh,
we can,
sorry.
we can continue.
Yeah.
All right. Zooming.
Okay.
We've gotten a lot of good shrimp shots today, though. I gotta say.
Yeah, this is a different species. It's paddling with the arms are going directly under the body and not out to the side so much. So actually, I'm not sure what this one is.
Stand by.
I'm afraid I'm just gonna have to stick to shrimp. Gonna
Yeah,
have to
maybe
send
a
a
bit
message
more.
to Mary Wickstein to see if we can get a little help from her on these arthropods again. The body doesn't look robust enough to go to something we tentatively identified as Canthophira, and it looks reminiscent
Okay,
of
taking
something
tiny
I
bit more.
can't remember the name of.
One last time.
But yeah.
Last time.
Okay. Thanks,
Nice.
Andy.
Thank you.
Appreciate it.
Nice follow.
It's a pretty good look at it, so hopefully we'll be able to identify it.
Okay, continuing on. Oh, what do we have here? Quick
Oh,
zoom
what's that
video.
interesting? Oh, what's that? Oh,
Oh, big polychaete. Wow.
wow, that thing's like 10 centimeters across.
Take a bit more.
Go ahead. Move back a little bit. I'm going to be moving away from it some more.
We've seen one of these before. I don't know what it's called, though.
Is that full zoom?
Well,
No, not.
Okay.
I'll take
we've seen
Give it a
them
try.
in really deep water, these big ones, but this is pretty shallow for one of these big ones, at least in my experience.
I think this is a polynoid scale
Oh,
worm,
he's out.
There we go.
similar to what in shallow water would be Aphrodite,
I'll
but
follow
it
him a
is a
little.
Okay.
Oh, wow.
scale worm for sure. So polychaete.
He's coming straight for
Oh, yeah, well,
Yeah.
certainly
Coming
polychaete.
So
We're just trying to figure out which.
yeah, polynoid maybe is the best we can do for the moment.
ROV nav bridge, move complete.
Yeah, definitely polynoid.
Funny.
Okay, he's in my camera somewhere. There he is.
I always love a good 10 centimeter. Oh, there we are. It's still there.
Coming
ROV
in.
nav bridge.
Good, Bridge.
Move is complete.
Move complete. Thank you, Bridge.
Are you guys going offline?
That'll
or
be a nice
it's
treat for
blinking?
Worm
I got a little
Wednesday.
blinking light
Yeah.
up here.
Worm Wednesday.
That's a Twitter thing. Every Wednesday, people show worms of
RTS.
different kinds all around the world.
Does
Hashtag Worm Wednesday.
that just means I'm keying you? I think so. Does ROV
Yeah, come
nav
on.
blink?
Yeah, it's blinking.
Okay, that just means that I keyed you. So
Roger. Okay, thank you.
Did you want to keep trying to watch these, or as we can follow?
I think we're done if video is done.
Okay.
Okay, let me just let him leave the frame.
So is there like a Sponge Saturday or anything?
Sponge Thursday.
Sponge Thursday.
Thanks.
Seriously, there is a
Thank you
Sponge
for that.
Thursday. Hashtag Sponge Thursday.
Were you all the way out?
Okay.
Yep, that's
the
full
ship
range.
just stopped moving.
Monday
So you
and
got
trial
about
by
a couple minutes to
Tuesday
swing.
and-
Great.
Yeah.
Coming
This
forward
is
some
what
more.
I'm missing by not being really up
About
on
20
social media.
No worries.
29 minutes left on the bottom.
29 minutes.
You know, in Hawaii, you don't say no worries. You go minor. That's
Yeah, but are you really missing out?
That's right. You guys want to incorporate a little Hawaiian wherever you are in the world. It's sort of, I guess, pigeon Hawaii. Then it's everybody. Somebody said something. Oh, I hope you aren't too upset about that. And then you go minor. So, anyway, just trying to provide a little extra.
Interesting.
The Cresa Gorgia that we just passed on the left. Unbranched bamboos on the right.
Was that? Did you want to image them or just continue flying?
Just continue flying, thanks. Just sort of calling things out
Okay.
for the. Maybe this coral that's that's a Trisopathes, I think. Yeah, we can. I think we imaged that one.
Okay.
We can keep going. And could we zoom on that coral that we just passed? Looks like there's a bunch of things on it. I wonder if we could take a quick
Is that
look.
right here?
Yeah, right there.
Sure. We get a little bit better footing here in case there's something.
This is going to be in the Primnoidy.
Come on in, video.
That is the coral.
Let me just hold and establishing really fast.
Okay.
Yeah, this is probably a Neurella
Okay,
in the genus Neurella in the family Primnoidy.
It's not quite full.
Okay, let's come out just a little bit.
Sure.
Right there. What do we want to look at in particular on here? Watch leads?
Some of Chris Ma's creatures there.
Okay.
Well, brittle stars, close, but
Close enough.
I'm happy to adopt some. Sure. These are Ophiocanthid brittle stars,
We can come on in now,
Ophiocanthidae.
Roger.
These are typical members of octocoral communities. I think they're mostly there as commensals. They take advantage of the water currents and use the coral for protection, perhaps, or just as a living, sort of a setting for their a substrate for living, and the spines protect them from predators and so forth, probably feeding on food as it comes by when the coral feeds. But we don't really know a lot about the relationship. They're not apparently predatory on the coral itself.
That's just commenting that you can sometimes tell primnoids from a distance because they host lots of ophiuroids. So, and I would agree. That's how I sort of first spotted this coral. Looking at a black coral, Bathopathes, another ophi on the seafloor there.
Looks like another. Is that another polychaete worm that's on the floor?
Sure, go ahead.
What that is?
No, it's some kind of thing, like a fin or something.
Those sort of look like a fish fin, doesn't it?
Yeah. But this is another Ophiacanthid. These might all be Ophioplinthaka. I'm not sure. I'll have to take a closer look at the disc later.
But that thing is probably not a fin. It's probably part of a shell or something. But I'm not really sure what that is.
That's next.
Are you happy here, Watchley?
Yeah.
All right.
We're comfortable with our lack of identification of what we were looking at.
Would you guys like another move, or
I can push out, sure.
onward?
Yep.
Bridge, ROV nav.
This is the bridge. Go ahead.
Yeah, we'd like another move, same as last. 30 meters, bearing 225
rock
degrees,
covered with
speed
those
0.2 knots.
demasponges, the piloderma that we imaged earlier.
Range 30 meters,
In addition
bearing
to
225
In
degrees,
addition
speed
to
0
bamboo
decimal
coral
Good copy.
and
Coming
dead
up.
sponge stock.
ROV nav, move initiated.
Move initiated. Thank you, Bridge.
Now, that's not a sea cucumber, is it?
Right by the lasers there?
Yeah. Or is it just a?
I don't know.
It's just a shadow of something. Maybe we ought to take a
rock,
peek at it.
maybe? I think it's a broken rock face.
Is that what it is? Oh,
We can do a
okay.
quick zoom. It looks like it is.
Yeah, it is.
Right,
I can see it.
is.
Yeah.
Never mind.
Would you like to zoom?
Okay.
No, we're good.
Thank you.
And there's an
That
Iridogorgia.
forest of bamboos to the right, and you're looking at broken rock.
Got to accommodate geologists too, Scott. But of course, we didn't think it was a rock when we zoomed it. Or I didn't, at least.
I would have believed that if you had called it rock at first.
So yeah, there's a nice little stand of bamboos. There's a really tall one in the back there. I wonder if we could get a better look at that. That thing's about 2 meters tall or something. Wow.
And I guess we should point out the black corals and Iridogorgia also, so a little bit of diversity in here.
Yeah. I'd just like to get an idea of how big this leggy coral is. It's getting up there. I don't even see the lasers now, but I think it's one of your really leggy ones that we imaged earlier, Scott, that had the 2 sub-branches off one side, but maybe I'm wrong. Maybe it's something different.
Yeah, it certainly has that overall morphology, doesn't it? I'd really like to know what the cue is that initiates branching in these guys.
And there's something on it, too.
It's like a brittle star, barely hanging on.
I don't want to get too close there.
Yeah, we're trying to
get some lasers on it for you.
So, yeah, Pilot, you think this thing is 2 meters, or how tall would you think this thing is?
Well, here's the lasers right at the base of it.
I would guess 2 meters, at least. It's taller than one of us, right?
I would think so.
Yeah.
Wow.
I'll go up. Yeah.
Yeah.
Yeah, definitely.
3 meters, maybe. Wow.
Jeez. Wow.
So Scott has often talked about some of the unbranched ones. Once they get old enough and big enough, then you start to see branches coming off it, and that may be exactly what we're looking at here.
Partial.
Do you want to get a close-up of the branching?
Yeah, right at the where it's branching there might be helpful for the identification. It's like it's nodal, maybe.
Perfect.
Yeah.
Not quite full.
All right, you're on, Scott.
Yeah, I'm having a look here. One of the things I can see, I'll point out right away, is that there are little white balls, very small balls at the base of some of these polyps, just along the skeleton. And those are likely eggs that are developing inside the polyps. They hang on to these eggs and may release them for fertilization, or may be picked up, or may pick up the sperm and do the fertilization within the polyps. I'm not too sure that we have much data on that in these bamboo corals. So we can see that there are a series of rods, not really long needles, but rods instead. And I think that this might be branching just past the node. So this would be technically an internodal brancher. And I wonder if this could be in the, well, it's hard to say, it could be in the Jasonisis group, but that's a really nice image that we can use for later, so thank you for that.
Okay, come on out, video, when you're ready.
Thank you, Scott.
Right there.
ROV nav bridge.
Yeah, thank you, Pilot. That was
Full width.
let's get the whatever's on it.
Get
really
a little bit of a
quickly
wind
if you
shift
can, that
to
red.
starboard. I'd like to
Oh, you want the,
yeah, the creature.
to about a 110 degrees.
Or cousin of the beasts.
That sounds okay. Is your center of rotation A-frame?
Let me double check.
Let's go ahead, video.
That is correct. Center of rotation.
And if you got some more, go ahead.
Okay, you can change your heading then. Thanks.
Yeah, this is Les at UH. There's a couple of really odd things about this coral. I was looking at the details of the sclerites in the polyps,
You got a little bit
Take
more.
a time.
which is what we used for trying to guess the genetic clade that these things are in.
Turn off the lasers.
From the imagery, and one of the things you can see is that the
Lasers?
basal part, or the lower half, the bottom half, of each of the polyps doesn't seem to have much in the way of the little rod-like sclerite. Sometimes there's a few small rods, but very often it doesn't seem like there's anything there. And what we've discovered over the last couple of years is that when we see a polyp like that, usually that means that there are these really flat, fairly thin scales in the lower half of the polyp, or there may be nothing at all. You can't really tell because the light doesn't come off the scale, doesn't bounce off the scales the way it bounces off the thicker rods. Even though the rods may be short, they're thick enough so that the light bounces off of them rather than goes through them. The second thing is that some of the polyps seem to have long rods between maybe four of the tentacles on one side, but not on the other side. And that we've always sort of used as an indicator for the clade C. But the problem is that clade C usually isn't branched. And so here we have a colony that's fairly sparsely branched and has an unusual sort of look to the polyps. So all in all, I would say this one's kind of a strange one in that if there's still an opportunity to collect anything, then a piece of this one might be rather interesting to get.
Let me see if I can get near it.
Well, I would go further left and say that clade C never branches, so that would be surprised if this were clade C. And this is the very sparse brancher that I've been pointing out for all of capstone. This is our third year of capstone, and every time I see this thing, I'm confused by it because it really looks like a whip that's branching, which doesn't make sense from the taxonomy.
Can you stand by for a moment, please?
Yes, standing by.
One other thing, Chris Ma, I wanted to point out, we had that close-up of the ophiuroid, and you could see it looked like an ophioclingthaca, perhaps, and where it was clutching onto the small branch, there was no tissue, and that's what I was trying to describe earlier when we were looking at the whip, that perhaps a crinoid or something like this was hanging on there, caused the tissue to be abraded, exposing the skeleton, but then it left. The tissue of the coral was able to regrow over that spot.
Like.
So you might want to set up closer.
He's telling me to stand by.
So gentlemen, the pilot, I just told the pilot if this is a value and you really think this is something different, what's the consensus here?
Why don't you set up as if you were doing a sample?
Coming in.
Let's see here.
I'd be thrilled to have a collection of this.
All right, Les certainly was asking for it too. I would support it. Can we get a piece of this pilot? We still have time
yes,
for
we
that?
can. I will. I'm trying to set up without crushing these others here, and I will grab you a sample.
I'm having a look at this, Chris, and I'm not sure that I can see a spot where you could get a branch.
So do you think it'd be better to take one floating?
Let's see, I guess it's actually a second colony. The one that had the brittle star on it, it might be possible to clip that here? Just below the branch point.
Do you think it'd be better taking one floating with everything that's around here?
That looks like the one that would give you the smallest piece.
I don't want to crush this, but that might be the only way to get close enough to float.
Yeah, I'm trying to think. Even that one with the brittle star behind it, that's quite a long piece. Unless there's another branch point behind it that I'm not seeing.
Yeah, I don't mean where the brittle star is. The branch is higher than that. I just mean that colony.
Yeah. Did you copy that, pilot? He was talking about the one behind this tall one. He was trying to figure out if there's a way to get a piece that also had a branch point in it.
Let me see from this angle back here.
Yeah, and I thought it was still pretty long. That's the only thing, Scott.
I don't think they're going to fit in our box.
Can you see this, Scott?
Okay. Yeah, I don't have a good sense of scale, so then that's fine. Just take a clipping.
Yeah, so
get 10 minutes on bottom.
I think they're 11. They think they're both the same thing, so you can just grab a piece from one of these branches that's easiest.
Okay. I could try right up here at the tip then.
Okay.
Get 10 minutes.
I don't think Joylock's
so
going to work
Les
here,
and
Andy.
Scott,
You
we
gotta use
need
auto
to
XY.
get a little clarification on what our field idea is going to be for the data on this collection. So you think it is now I've forgotten, a possible sea clade, is that correct?
Les had suggested that it has characteristics
gonna
of both
drift,
the C
so
clade
I
and
just want
the
to make
D
sure I'm not
clade, so
taking
it's
off
a
and
little
it
bit
looks like
hard to tell.
in a little bit.
Give me just a second, Chris.
Okay, thank you. It's going to be probably starboard inboard, I would think, if I'm not mistaken, but we can change it if they change it. He's taken a second. Yeah, how about acidity or yeah, here he is. Okay. Yeah, go ahead, Scott.
Yeah, so I would call it sparse branch D.
All right, am I spooled up?
All
Yep.
the C clade ones that I see have very long intertentacular spines, and this one does not. So let's play it safe that way.
So sparse branch B clade, you said, right? You faded just as you were giving the letter designation for the clade.
Yeah, D as in dog.
D. Okay, got it. D. So sparse branch. We got it. Thanks, Scott. Yeah, that's correct.
I'm going to bump your auto XY.
Yes, please. Thank you.
Perfect. Good job.
Back you up.
Thank you.
Let me know when you're ready for the draw.
Okay, go ahead. It's a good thing you had me test with those blocks the other day.
Yep. Nope, you got in. Just let go. There you go.
It's in the box.
Okay, go ahead.
Great job, pilot. Thanks, guys.
Thanks for that, everybody.
Thank you, Scott.
Yeah, thank you.
Good job, Andy.
How much time we have left?
6 minutes. 1:30 on the nose. You guys can head up.
Hydraulic secure here. Okay. You said 6 minutes left?
Yep.
So, yeah, no, this is specimen 5, right? Okay. Few notes for the dive summary report tonight. Okay. So we got a little bit of time, probably about 5 or 10 minutes if I'm not mistaken. And that completes our collections for today. Pretty much filled the boxes. So we can make a run for it.
Just FYI, you're putting a turn in. Or maybe
Am I
not.
taking it out? I thought I was taking it out.
Oh, another polychaete.
What was our previous pass again? It was 220.
Yeah.
Another big one, or
225.
Yeah, over there.
oh yeah, there is another big one. Wow.
Oh, this guy?
Yeah, or is that a
Oh, it's
is
a
that a
cucumber.
cucumber? I was about to say, yeah, if we could get an image of this.
You still have
That's
me in
interesting. They're colored very similarly.
I'm not going to tell anybody that you thought it was a worm. Honest.
They're colored similarly.
yeah,
that's
yeah.
that's. Wow. So, I think we've mentioned this before. This is Hansenothuria.
Come on, video.
But what's really dramatic about this is the clear body wall. You can literally see its gut filled with sediment
Partial.
castings, just about ready
That's
to
perfect, thanks.
to be released. So
Unless you want something
the
more.
mouth on the top side digests organic
was that
material
Whatever
along
shot
with
you'd like.
a bunch of
Roger,
sediment,
okay.
sand, mud, whatever. This all goes into the intestine. There you can see there's a long
Alright, I'll
sort
just
of
get
the
a
stomach,
bit of a
the
closer
forward intestine, and then more of a long
zoom.
intestine towards the rear, sort of looping over itself back and forth. And then when it's ready to
Just
go,
tilts
it'll
up
poop all
slowly.
that
We
stuff
could,
out.
kind of
Or sometimes they'll
scan
swim
the animal.
and dump all that stuff out before they go. So the
back down.
Coming down.
the D2 is making a very fine video pass so that we can see the track of this animal's dinner, probably breakfast and lunch too. And you can see the sediment right there. And those are clean grains. That's actually, believe it or not, there's actually an ecologically important function to that because,
All right,
at least
come on
in
out.
shallow water species, they aerate the sediment, and that permits other animals to live in there and to exploit the substratum. So
Right,
sea
2 more
cucumbers
minutes
function
and
in
you guys
an
can start
important
packing up.
ecological
Sound
sense.
good?
Chris, some of the
Don't
shallow
see
water
anything
studies
out
have
there.
also shown that
You guys are going
deposit
to
meters
get choke
change the
configuration
microbial
and go up to
community.
900 meters.
So the sediment
30 meters a minute.
before it gets eaten and the sediment after it's pooped out has a different suite of bacteria.
Oh yes, that's right. So even though I love to comment on sea cucumber poop, and I've written a blog about all of the important aspects of it, there really is a lot of important ecological function to how these animals live. So they may look weird, but they contribute in their own way to the broader ecosystem.
We've got a stock chrysogorgid with another fair-nematid sponge. This looks like something we've seen before. Got a Bathypathes on the left, some more of those toothbrush sponges, the Piloderma. So we are kind of coming to an asymptote on the number of animals, at least at this particular part of this particular seamount, at least over the distance we've covered. We're seeing a fair number of the same things, occasionally getting something different.
Okay, pods, you guys can start packing up.
And one of the things that's interesting about today's dive, we've seen a number of
Configuration,
species, but there are a
it's
number
time.
of species that we haven't seen that we might
Zero turns.
expect to see in this depth
Bringing all
range.
the swing arms here. While he's doing
Number of
that.
other
We can
Watch
Gorges,
these, we're
Alligorges,
packing up and going to head up. This is our last
Carnarchians,
site of
so
quick
bottom.
zoom video.
a little bit deep for them.
Roger that. Thank you,
The
Pilot.
Primella diversity hasn't been all that high.
That's partial.
Okay.
Yeah, thanks, Scott. By the
All
way,
right, come on
thanks
out.
for suggesting this as a location. I thought it was a really, really beautiful dive. Got a lot of different animals. They are heading off the bottom now, and we're going to switch over to mid-water mode. And just a very quick wrap-up. We got 2 rocks samples, and we got 3 biologics. We got a sponge, and we got, let's see, we got his bamboo, and what was the other one?
We think we got one of the black
Oh,
corals.
the black corals, that's what it was. We got, senior moment. And beautiful close-ups. Videographers and the pilots did a fabulous job getting some amazing shots. We got into a moderate community. I would describe this, where some places were a little sparse, and then some places had some really nice stands. Scott, you got any other comments about this site that you'd like to make as we're lifting off bottom?
Well, only that it'll take some time for us to look more closely and try to quantify the diversity and the abundance to be able to compare to some of the things that we saw in 2015 and what we'll see on the next set of dives going forward over the next couple of weeks to see, is there any difference that we can discern from just the snapshot between the north side and the south side, and then, of course, at different depths. But I think you summed it pretty well. We saw some interesting diversity. Clearly, there were local high points that were really important. We saw a number of very large colonies, which suggests that the area has been stable for a very long period of time. The corals were probably pretty old. And a number of associates, organisms that are living with those corals that sort of prove the fact again that the corals and the sponges themselves create habitat and so increase the overall biodiversity. So thanks everybody for an interesting dive.
Thank you, Scott.
Thank you. And none of you people